Well, welcome everybody to the CRISPR in Agriculture session here. I'm Brian Staskawicz. I'm in the Department of Plant and Microbial Biology, University of California at Berkeley, and also part of the same institute as Jennifer Doudna, where I'm the Scientific Director of Agricultural Genomics. Today we're going to have 3 talks. I'll be starting my own talk, and then we're going to be going to gene editing in animals, but also then we're going to end up with gene editing for herbicide resistance. So the major talk here is genome editing. Go back to the first one, sorry. Genome editing is an advanced breeding tool for the sustainable production of crops. Next, please. I just wanted to remind everybody the research goals for the IGI Agricultural Genomics. We're interested in developing crops that are safer, healthier, and more nutritious to address human disease and malnutrition. We're developing high throughput, precise genome editing technologies and tissue culture regeneration in advanced breeding lines in crops like wheat, rice, tomato, cassava, and cacao. We're deploying genome editing technologies in these crops to significantly increase yields in an environmentally sustainable manner to address global food securities. And finally, we plan to collaborate with various CGIAR centers to be able to release our edited plants to the developing world. Next slide, please. So this is a great review that comes out of Kaishar Gao's lab, and really what it's emphasizing here is the developmental timeline for introducing traits into agricultural crops. So classically, if we look at just breeding, it typically takes 10 years or more to actually introgress a trait from a wild species into a domesticated crop. Other types of breeding have been transgene breeding, which are GMOs, which we know can take 8 to 12 years just because of the long regulatory process. And then also scientists over the years have used mutation breeding to introduce traits into crops in a very uncontrolled manner. But with genome editing, we can greatly reduce the time that's necessary in order these traits, because we're starting with an elite variety, and then we can precisely make particular mutations. Or we can do allele replacements. We can do all kinds of alterations in gene expression patterns. And so really the power of CRISPR technology in agriculture is quite enormous. You've heard a lot of talks today on its role in biomedicine, where it's going to affect hundreds of thousands of people. Eventually, CRISPR technology in agriculture will affect billions of people. So keep that in mind. Next slide, please. So I'm going to give an example today from work that we're doing in tomato. And what we're trying to do is to create durable and sustainable disease resistance to this important bacterial disease of tomato. Next slide, please. And just as for an example here, this is very simple and straightforward. Our gene editing approaches for controlling bacterial spot of disease of tomato is essentially identifying previously characterized mutants in other plants that provide bacterial disease resistance. For instance, I'll give the example of the DMR6 gene in Arabidopsis thaliana and the BS5 gene in pepper. The idea is to find targets that have already been established that when mutated give rise to disease resistance. And the idea is to identify orthologs in tomato of those particular genes and to specifically create CRISPR-induced mutations in those genes. So next slide, please. The DMR6 stands for downy mildew resistance gene. It was first identified in Arabidopsis, and its inactivation leads to broad spectrum disease resistance. It turns out that these mutants accumulate salicylic acid, which is important for disease resistance, and they're resistant to both bacterial pathogens, and you'll see other classes of pathogen. Next slide, please. So the most important thing for CRISPR technology for applications in agriculture is that you should have well-sequenced genomes of the target crop that you're interested in. So tomato is a great model system that we're able to actually identify orthologs. So over in the right-hand side, you'll see what's in red, the red boxes there. I don't know if you can point to those. There's 2 genes in tomato called Solanum glycopersicum, DMR6-1 and DMR6-2 in that particular case. So we identified those genes, and I'm not going to go into the specifics. Go ahead, keep going, the next slide. So what we did was essentially design guide RNAs, and we used Agrobacterium tumefaciens to introduce CRISPR-Cas9 with the guide RNAs, and then we identified targets. On the next slide. And just to make a long story short, we are able to actually get resistance to a broad spectrum of pathogens, including Pseudomonas, Xanthomonas, and Phytophthora in this particular case, and also powdery mildew. So you get broad spectrum resistance. In the next slide. So the second gene that we're interested in mutating by CRISPR in tomato is a gene that was identified in pepper. It's called the BS5 gene. Next slide, please. And this gene was cloned by map-based cloning, and it's a very strange gene. It has essentially 92 amino acids, and the mutation is an in-frame double leucine deletion in the C-terminal portion of the pepper gene. So next slide, we use a similar strategy. Next slide, please. We knew the sequence in pepper. Next slide. And then we could find the orthologs in tomato. We can click the next slide, please. And one more. There's the mutation. So there are really good orthologs in tomato. So very similar to what we did previously is we now design guides. Next slide. So what you're seeing in the bottom below is the two orthologs of the gene there, 98300, 98310, and we designed guides. Next slide, please. And again, putting in Agrobacterium tumefaciens and going through the transformation. We have a very robust transformation system in tomato. We can easily introduce the editing components and be able to target, and then we screen for mutations in the targets. Next slide, please. And we got several lines and where we essentially get one base pair insertions or we get deletions. And these mutations essentially have premature stop codons. And so we get a mutation in the protein that's encoded by those genes. Next slide, please. And you can see here's an example of an edited tomato line. On the left side is wild type, where you can see these little dark, greasy bacterial spot lesions. And on the right-hand side, it's lesion-free. And when we do bacterial growth curves on those particular plants, we find that they are greatly reduced by 1 to 2 orders of magnitude. Next slide, please. Another approach for disease resistance is to identify disease resistance genes from wild species. In this case, it's going to be from Nicotiana that recognize bacterial effector proteins. Next slide, please. And here is what I want you to take home from this particular slide here is that there's 2 layers of plant immunity. There's pattern recognition receptors, but also there are NLR, nucleotide binding leucine-rich repeat intracellular immune receptors in this particular case. And this turns out that ROC1 is one of these genes. Next slide, please. So we knew that this type of resistance was dependent on a downstream gene in the pathway called EDS1. Next slide, please. And what we did, so another really important component of CRISPR technology in agriculture is to actually make mutations in genes. So if you want to know the phenotype of a particular mutation, we can easily make mutations. So we made CRISPR-induced mutants in this EDS1 gene. Next slide, please. And then we had to identify the immune receptor. And I'm not going to go into the details here, but essentially what we did, we were able to use a combination of computational biology and viral gene silencing to identify putative genes. And then by going in and actually doing RNAi, we're able to identify the resistance gene called ROC1. Next slide, please. Now, here's the really important thing that this is really exciting research right now. What we wanted to do next was to be able to isolate the protein complex of the immune receptor, recognizing the bacterial effector. And to do this, we had to use this EDS1 CRISPR-induced mutant because we could co-express the NLR gene along with the effector gene and get high levels of protein expression. And then Raul Martin, a graduate student in Eva Nogales' lab, in collaboration with Zhong Qi in my lab, was able to actually purify complexes. Next slide. So what we found here by using cryo-electron microscopy, we were able to solve this structure at the 3.8 angstrom level. And what we saw was when the immune receptor recognizes the bacterial protein, it induces a oligomerization of this complex with these various domains. And then the next slide, please. So going from left to right, what happens is that the leucine-rich repeat binds the effector protein. In the presence of ATP, it induces a ROC1 oligomerization into this tetramer. And what's important now is that there's a toll interleukin-1 receptor domain at the N-terminus of this particular protein, and it brings these components into a specific configuration that now becomes enzymatically active for NADase. And we now know that NADase cleavage is important for signaling downstream. So this is the second example of a resistosome that's been isolated from a plant. Next slide. Now, what's exciting about this, which we never could have predicted, is at the C-terminus of this leucine, oops, go back one. At the C-terminus of this leucine-rich repeat, there's a what's called a post-leucine-rich repeat. And it turns out that we never could have predicted the particular structure here, but it actually has immunoglobulin fold, where it has a constant and a variable domain. The variable domain goes into the ZOPQ protein and specifically binds it. So what's exciting about this now sets up the example where we can actually start to use synthetic biology to be able to identify and make new novel receptors with this particular receptor in this particular case. The idea is once we are able to actually do that, we then can use CRISPR editing technology to introduce precise mutations into already proteins that actually give rise to resistance. So we're really excited about the possibility of being able to actually get novel immune receptors by combining CRISPR technology along with synthetic biology. And I think there's 2 more slides. So just as an example here, there are many approaches in agriculture. I only gave you a couple of examples today of our research in tomato that we're able to actually identify the core receptor ZOPQ, the effectors. We're able to use CRISPR-Cas9 to construct precise mutants and recessive alleles, and a combination of pattern recognition receptors and multiple R genes, along with novel receptors. We hope to be able to actually stack in various levels of disease resistance into tomatoes, so it would be very difficult for the pathogen to overcome as you put a selection pressure. So finally, I have a acknowledgement slide. So I'd like to acknowledge a bunch of people at UC Berkeley, Arturo, Daniela, Doug, Raul Martin, and Chung did the work in Eva Nogales' lab with the resistosome, and Farong and Miles helped in the protein expression. All this work was done at the Innovative Genomics Institute. Also, I'd like to acknowledge Alex Skoltink, who cloned the ROC1 gene, and he's an entrepreneur fellow at the IGI, where he started a company called Fortifite, and also funding from the USDA in 2 Blades and the Innovative Genomics Institute. So I'll stop there and be happy to answer any questions. Do resistant tomato lines show any decrease in fruit production? We've just started to begin the field trials with these particular constructs, and so we don't have, the DMR6 might be a little stunted, so we're still following that up. The BS5, we don't see any obvious changes in size. The slides will not be made available, unfortunately, because the last part is an embargo for science, where that article's been accepted. Can we use the same approach? Whoop, they keep going fast. What are the challenges in implementing CRISPR-Cas9 technology in crops regulatory approval? That essentially matters where you are. In the US, APHIS, Animal Plant Health Inspection Service, will allow simple mutations, deletions, insertions, which will not be regulated as a GMO. Europe, as you know, has much stricter regulations, and these are also being worked out in other parts of the world. In Latin America, we're starting to see that they may have similar adoptions that we've seen in the US. There may be some differences. Australia, Japan, This is all still being worked out with regulatory approval, but we think that it's going to be a great way moving forward, that we're going to be able to use CRISPR technology to actually make mutations in high-yielding crops, and not to be able to worry about doing classical breeding where linkage drag comes in. So, any more questions?
I think we can take one more question really quick, and then we can move to the next speaker.
How long will it take for any of your resistant lines to hit the market? That depends on who will actually license this technology and whether they're interested in implementing it. We don't know. It's probably several years out still. So I think just for time now, we're in 20 minutes. And so now we're going to move to the next speaker, which I think she's going to be able to share her screen. So that will be a novel concept. So it's a great pleasure to introduce Alison Van Eenennaam from the University of California, Cooperative Extension Specialist. And many of you have probably heard her speak before. I have. I've always been impressed with the great talks that she gives. And she's going to talk about genome editing applications in animals. Okay, Alison, take it away.
Thank you so much, Brian, and thank you very much for including animal in agriculture. We often do these things and only talk about plants. So on behalf of the animal kingdom, we appreciate being included. So thanks to Synthego for inviting me. And I'm going to jump right in and talk about the fact that breeders, be they plant or animal, have always been selecting on alterations in the genome. That's the basis of evolution and selective breeding, and we can make dramatic changes to our species, be they the wolf in this case, to our different dog breeds, or more importantly in this context of this talk is agricultural alterations. And shown on the screen here is the changes in poultry from 1957 to 2005 at different ages, and you can see the tremendous fourfold increase in the rate of growth to get to market weight. And a similar story could also be shown for example, the dairy cows, where the yield per milk cow has increased more than fourfold since the 1940s. And we can see this inflection point here in increased yield as the result of an adoption of a breeding method. So for you tech nerds, this is not artificial intelligence. This is artificial insemination that enables the best use of the best males and has basically resulted in a decrease, listen to me, a decrease in the number of dairy cattle in the US from 27 or so million in 1940s to 9 million today. And so I think if you're interested in the environmental footprint of agricultural production, breeders are your best friends. And I just showed this that shows with regards to dairy production relative to 1944, each cow produces more than fourfold what she did in the 1940s. And as a result, there's less animals, less feed, less water, less land, less manure. And the bottom line is the greenhouse gas emissions associated with a glass of milk today is 1/3 of what it was in the 1940s. And I think breeding is such an important aspect of sustainability. So now let's ask, are we going to be able to use genome editing as a breeding method to improve our animals? And unlike plants who have to use agrobacterium to introduce editing reagents into their species, we're typically, at least in my lab, focused on the one-cell zygote. And so I know that germline editing is a controversial topic in humans, but from an animal breeder's perspective, if you're not germline editing, then what are you doing? And so we very much are trying to edit this one-cell embryo just after the fertilization. And our ultimate game is this creature here, the homozygous, non-mosaic altered animal. And the alteration in this case might be a knockout, and we would like both of the chromosomes to be knocked out. And then it might also be a knock-in. And ideally, we'd like both of the homologs to be knocked in also. And so we try to get that edit done quickly before that first cell division that leads to the cells starting to multiply to form a zygote and ultimately an embryo. So what might we knock out if we're looking at animal breeding specifically? Well, you might knock out genes associated with allergens in animal products. Or you might knock out genes that make animals susceptible to disease, not unlike the plant scenario we just heard about. Or you might knock out genes that result in unwanted development. And I'm just going to go through some examples of extant applications, not commercialized, but reduced to practice that have been in the public literature. This one is a 2016 example of a genome-edited pig where they knocked out a gene that resulted in those pigs no longer being susceptible to a disease called porcine respiratory and reproductive syndrome virus. Did I catch your attention with the respiratory virus? If I didn't, I'm not really sure what planet you're living on. This is a really problematic disease. It's about a $600 million a year loss in the US and 1.2 billion in the EU. And this is actually a product that is being taken through regulatory by a company called Genus. And I realize I'm a geneticist, but to me, genetic improvement to address disease challenges is a better solution than having those animals get sick and then needing to be treated with antibiotics and chemicals. And so I would argue it's a much more sustainable approach than treating sick animals. Similarly, a group in New Zealand, in this case, in collaboration with Recombinetics, knocked out the gene that encodes for beta-lactoglobulin, which is one of the major allergens in milk. And this cow, conceptually, could produce non-allergenic milk. Similarly, there are groups working to knock out genes associated with maturation, in this case in pigs. Pig castration is an invasive and frowned upon procedure with increasing animal welfare concerns. And so there are groups that are working to basically take out the gene that results in, if you will, sexual maturity in pigs. So they're in a kind of stay in a prepubescent state so that they don't need to be castrated, but can still be consumed without getting the attractively named boar taint associated with their pork. There are also some really interesting applications that don't have to do necessarily with a single gene knockout, but things such as surrogate sire technology, which was recently quite a big paper in PNAS that came out. The idea is that you'd like this genetics. This is the boar you would like your genetics from, but you're going to actually use genome editing to ablate the germline of this animal, so he no longer will form fertile spermatogonia. And you're going to take the spermatogonial stem cells from this animal and populate this boar's testes so that when he's mated to a sow of the breed of this boar, you get a purebred line here. It's artificial insemination on legs, if you will. And that has a real potential to really change breeding program design. Those are some of the things you could do with knockouts. What might we do with knock-ins? Well, we might knock in alleles that make them disease resistant. We might knock in alleles that take away unwanted development, or we might knock in useful genetic variation. There are examples where a group in China, in this case, have knocked in an allele that results in these cows no longer being susceptible to tuberculosis, which is quite a big problem globally and is actually quite a spread going on in the UK. As a result, actually, of spread by badgers. And so there's an interesting kind of fight between the badger lovers and the TB haters, if you will, as to whose rights prevail. And so I think that's an interesting one. You've probably heard of the hornless animals. So horns are removed from dairy animals typically to protect them and their handlers. And in this case, you can introduce an allele from one breed, such as Angus, into a dairy cattle background, and this was done by the company Recombinetics, and result in animals that no longer need to be manually dehorned because they're genetically dehorned. And you may have heard, and this is an example from my own lab, and this is a 5-year project of my PhD student, Joseph Owen, who was trying to knock in the SRY gene that triggers animals down the male differentiation pathway. He was trying to use CRISPR-Cas9 to knock in one-cell zygotes. And one of the big issues in one-cell zygotes is that the HDR pathway is pretty much quiescent, and the main repair pathway is the NHEJ pathway. And so he used some ideas to basically knock in the SRY gene onto an autosome, in this case chromosome 17. And this is a heterozygous bull who has the normal Y chromosomes that he would pass on to his offsprings. And when mated with a female, you're going to get males, fertile males. But then he's heterozygous for the SRY gene on chromosome 17. So in this case, you would just get a male that's fertile. But what we're trying to do here is skew the gender ratio or the sex ratio. And so this XX would give you a normal fertile female, But in this case, the SRY on the chromosome 17, when combined with the X chromosome from this bull, would give you basically a phenotypic appearing male. And males are more efficient at converting feed to gain and are the desired sex when it comes to beef cattle production. And so there are all sorts of things being done in cattle breeding programs, but I guess I want to kind of point out the fact that our objectives haven't changed that much. So we'd still like to increase product yield. We'd like to address welfare concerns. We'd like to help adapt animals to a changing environment, eliminate milk allergens, introduce disease resistance. And in the last two examples I gave you, alter breeding program design by either increased males or perhaps increased superior genetics in the surrogate sire approach. So our overall goals haven't changed that much. I kind of see editing as a cherry on top of the breeding sundae. That is, we're going to keep using genomic selection and performance recording and progeny testing and embryo transfer, all of the things that make up a normal breeding program. Genome editing allows us to targetedly introduce useful novel genetic variation without linkage drag. That is, I don't need to bring in all the genes from a breed that I don't want. I just introduce the allele that gives me, for example, not growing horned phenotype. Will we be able to do this? And if you're interested, this was written about in the CRISPR journal back in 2018. I have an article there about gene editing regulations in animals. And I'm just going to kind of contrast the difference in the United States as it relates to plants, as was just discussed by Brian, where if you introduce an edit that could have been developed through traditional breeding techniques, then you're not going to be subject to regulation as a GMO, as distinct from animals, which are regulated by the FDA, where if you used editing and you introduced an alteration intentionally, including nucleotide insertions, substitutions, and deletions, you're going to be regulated as a mandatory new animal drug. This is a mandatory regulation, and no animal can go to market without this mandatory evaluation. And we have this really strange dichotomy between the two kingdoms now as it relates to regulations that's going to make it difficult for animal breeders to use this technology. If we look globally, the situation is kind of mixed. So a gene-edited knockout food animal would be subject to additional regulations in the EU, New Zealand, and America, but not Japan, Canada, Brazil, Australia, or Argentina. So that's a knockout. A knock-in of an endogenous allele would be subject to additional regulations in Australia because they're using the use of a donor template as their trigger, and additionally the EU, New Zealand, and the United States. So we have this very disharmonious regulatory approach, which I think is potentially really going to limit the ability of animal breeders to use this technology, because there's obvious trade implications if we have differential regulations for things like knockouts and deletions. And I find it kind of difficult to reconcile as a geneticist how a SNP or an indel can be a new animal drug, because if that's true, then I would argue that we're all high because we are basically a mixture of mutations. That's why we all look different, and that is the very basis of evolution. So I will stop there, and I'm very happy to take questions.
One question was, do you think the increase of production through genome editing will plateau, or is there still a huge potential that exists in this field for the future?
That's a good question. What's been intriguing in the long-term experiments with increased maize yield and also dairy cattle selection is that we keep thinking we're going to plateau out, and we keep cranking along at pretty much the same rate. So I think that probably conventional breeding is going to be where we get a lot of the continued benefits in our quantitative or multigenic traits. Where gene editing comes into me is the ability to introduce useful alleles from maybe a less desirable breed into a more desirable breed, or maybe introduce something like disease resistance into an optimally suited breed. So it's the absence of linkage drag is where the real benefit of genome editing is in breeding programs. And so I want to emphasize, traditional breeding programs are not going to go away. This is a I guess, an additive or it helps the conventional breeding programs.
Great. One more question we have. Will knocking out the unwanted phenotype cause side effects to the animal? Do you see any side effects when you?
That's a very broad question. It kind of depends on what phenotype are we talking about, I guess. And so, for example, in the case of knocking out horns, that mutation doesn't appear to be associated with any other phenotypes in the animal that's been around for several centuries now. But obviously, if you knock out an essential gene, you actually just won't get an animal survive. And so I think it depends what it is that you're going to be editing as to what the potential side effects would be. And that's true for any animal breeding program. You know, sometimes you select for more milk, you might decrease fertility. So you always want to keep your eye on correlated traits and make sure that you're not going in the wrong direction.
Great. We'll take a few more. We have about 5 minutes. Noah asks, has there been any work on longevity in farm animals? Seems like there is an incentive and could ladder up to humans.
Not really. So, I mean, I'd hate to be a bit callous about it, but what we're trying to do is get our animals to market in as short a time as possible so that they're consuming as little feed as possible. And so the only place longevity might play out would be in breeding stock, perhaps, where you might want, for example, a cow to have multiple calves. But because genetic improvement is kind of cranking along at the rate it does, older animals, by definition, become less genetically superior. And so there's kind of a cost-benefit balance there where ideally you'd like all of your animals to be the most recent generation because they're going to be the best genetically. But then there's some costs associated with developing the animal and getting them to reproductive age. And so somewhere in the middle there's an optimum. And so we're not really selecting for animals that can live a long time because typically agricultural animals don't live out their lifespan. That's not their purpose. So I'm not really sure that that's really something that's being selected on. Certainly, we would like animals that are fertile and have a calf every year. So that's a very big selection objective. But longevity is going to be depending. It's going to have that balance as to which animal, what age distribution in your herd or flock is optimal in terms of production, and then also repaying the cost of developing young animals.
Great. And then we'll take one last question. Jennifer asks, have you tried any dCas9 base editors for embryo genome editing since it's less HDR repair, since there is less HDR repair?
No, we haven't. I'm certainly open to that. I guess that if we're talking about base editing, I'd have to look into that to understand exactly how that might work in embryos, but I'll look into that.
Okay, great. I think we can move on to our next speaker.
Okay, so I'll introduce the next speaker then. Great. So our final talk this afternoon, is from Juan Fernandez, who's going to be talking about empowering crops for weed control using CRISPR. Okay, Juan?
Hello, can you hear me right?
Yeah.
Okay, there you go. So thanks everybody for being here. And thanks, Synthego and Kevin for the invitation. Today, I'm going to talk how we at Bioheuris are empowering crops for weed control using this fantastic tool, CRISPR. So weeds have always been a major concern for farmers and agriculture in general, because they basically compete with small resources like water, nutrients, and light with crops. And this could cause up to 50% decrease in yield production. Herbicides so far are the best tools to control the weeds. But unfortunately, the sustained use of those herbicides, like a limited number of herbicides, has led to the emergence of different weeds biotypes that are basically causing a huge I don't want to call disaster, but it's causing a huge difference in weed management cost. For example, here you can see how in the US and Argentina has basically doubled the cost in a soybean field in the last 10 years. And now we can imagine that this is not only for soybean, but also for another different crops in many other different countries. So taking this in consideration, what we are doing at Bioheuris is developing a sustainable weed management system that will bring farmers a new generation of multiple herbicide-resistant traits that will certainly delay weed resistance, will reduce the weed management cost, and of course, time of management. And the idea is to use a herbicide that has less environmental impact. Even though at Bioheuris we have two main platforms, one that does gene discovery in a way that we can identify new potential targets with new mode of actions. The other platform is the one that I'm going to be talking today. And I'm going to be telling you about our efforts on how we are building this platform in a way that we speed up the natural evolution process of crop, comparing with, so the idea is to evolve the crops faster than the weeds. And how do we do this? So the platform basically, so you can imagine an engine with two wheels. So one wheel is the plant tissue culture, and the other wheel is the genome editing, right? So in this case, we use soybean and sorghum, and the delivery method we are using here is gene gun, but in this case with very fancy bullets. Those bullets are the ribonucleoproteins, Cas9 or CPF1 or Cas12a. But before doing this, before loading the gun and shooting the explants, so we are doing the in vitro validation. And in this case, we are using, as I mentioned before, either Cas9 or Cas12a. And the guide RNA we are using are from Synthego. And actually, this was a great results in our hands, because it not only increases the efficiency, but it also allows us to reduce the time of production and the production and checking of the in vitro activity of the guide RNAs. So okay, then we have those guide RNAs already validated, the RNP already validated. So we load the gun, and then we shoot the meristem. The idea is to generate, of course, as you know, so the double-strand break, and then to ultimately introduce a precise modification in the genome in a way that those cells or those plants that receive that precise modification will be resistant, comparing with the other ones that are, for example, incomplete for the homology direct repair, or they have a wild-type DNA. You might see here like a pretty straightforward, in this drawing, you might see a pretty straightforward set of steps, but actually every single step, it requires a lot of optimization. So probably you're not, if many of you are not familiar with gene gun, I'm going to show you how a typical pattern of gene gun looks like. So first thing we did was we set the conditions, like the physical condition for gene gun bombardment in soybean and sorghum explant. And in this case, you can see how, so we use reporter genes in this case. So, and here, so you can see the transient expression in soybean meristem. Those blue foci that are expressing the gene marker, the gene reporter, and also on your right side, so you can see how the sorghum callus looks like after gene gun bombardment. So what you're seeing here is the typical scatter pattern from gene gun, which is basically you have in the background you have millions of cells that did not receive any impact, and then a few thousand that did receive the impact. So encouraged by these results, what I wanted to do is, okay, let's move to the next step, which is, so we set the parameters, so let's go and try to actually do the double-strand break, to induce the double-strand break. And to do so, so what we did was we tried several conditions. So here I'm summarizing just some of them. So like a microparticle preparation protocols, there are like many different, well, not that many, but few microparticle preparation protocols. The guide RNA source, as I mentioned before. So we did the IVT one in-house, and also the guide RNA from Synthego, and the difference was way better using the Synthego guide RNA. So thank you guys. We also the target site. We pick different target site. transfection region. So there are some different protocols that use different transfection regions. The molar ratio between all the components of a microparticle, of that fancy bullet that I would like to call fancy bullets, and then different germplasm. And the results are for soybean and sorghum pretty encouraging, because we so you can see here in the x-axis numbers, but you can imagine those numbers as a mixture of different a range of different conditions, different factors that I mentioned before. So we identified those factors, and we basically went through different to several rounds of optimization until we end up comparing our numbers with the numbers that are out there. Like, for example, what is the percentage, by using the same, of course, approach, like the ribonucleoprotein bialistic, what is the result that other groups have gotten? So in case of wheat and corn, they have 0.21 and almost 0.7 percentage of indel using ribonucleoprotein. And in our hands, in soybean, we got 0.5 and 0.21, which is pretty comparable with the results from other groups. So this basically was encouraged for us, and then we decided to move forward and go ahead and try the proof of concept. For the proof of concept, what we did was we target the ALS gene, both in soybean and sorghum. That gene, it has been described that precise modification in one codon, the tryptophan 574, if you change that tryptophan for a leucine, so the plant or the crop is resistant to ALS herbicide family. So first thing we did is we designed many guide RNAs. We tried different nucleases, as I mentioned before, Cas9 and Cas12, and then we tried different germplasm. And actually applying those conditions before for the GenGAN setup, and then those conditions, those optimization from the double-strand break, we also identify condition here for inducing double-strand break. This is very important because it's in a DNA-free fashion, because our bullets are just RNPs. So we identified those conditions and decided to move forward to the ultimate goal, which is basically produce this precise modification in that particular position. And what we did was we basically mixed that RNP, that specific guide RNA, with that specific protein, mix it together with the repair template, and we tried several strategies too. So like the DNA repair mass, the guide RNA, of course, that I mentioned before, and also the germplasm. And what we see was pretty good, because we saw homologous direct repair induced in vivo in our elite lines, soybean and sorghum. That was pretty great, actually. And we did it without double-stranded DNA. So this is actually when we did a close view of these results, we actually compare Cas9 and Cas12a, because when I was doing my postdoc at Yale University, I did work in optimization of Cas12a or CPF1. And then we decided to also try the CPF1 strategy here to see, like, for example, in zebrafish is also producing a higher HDR frequency if you compare with Cas9. And actually, that was what happened in our case. So where we compare elite line, in this case in sorghum, with the public, and we have gotten higher HDR frequency by using CPF1 comparing with Cas9. So we are pretty excited and happy about these results. So moving forward, what we are doing now here is basically doing the massive bombardment. So we are bombarding several hundred different explants from soybean and sorghum, and then exposing them to the specific herbicide. And then those plants, of course, that are that went through the precise modification will grow, and the ones that are not modified will basically be sensitive to the herbicide and won't survive. So the message that I want you to take is that we developed this platform that basically combines this plant tissue culture, which is not trivial because you have to set up all the conditions and all the protocols to basically raise or in vitro culture your elite lines in the lab, which is a huge milestone. And also we combine that with the gene editing. We demonstrate the activity of Cas9 and Cas12 in elite germplasm, which is pretty great actually, because now breeders, so we have a really, so we have partners, different breeders here in Argentina and also in other countries, where they basically provide us with elite germplasm and we can potentially do the job. It's not only that we have shown the double strand break that is working, the double strand break, but also we have shown that we can introgress precise herbicide-resistant trait in a no-GMO manner, right? So because we basically do that RMP, we combine it with single-strand DNA oligo, and then we induce that precise modification. And with this, I would like to thank the whole Bioheuris team. This picture is a little bit old now because we have more people in the lab, but especially I would like to thank the genome editing platform, Maxi, Monica, Belen, Luisina, and Cecilia. And with that, I will be happy to take any question. Thank you.
Okay, great. We have a few questions for you. There you go. Let's see. Does the low transformation efficiency of plants like sorghum make it difficult to use CRISPR?
Well, actually, what is difficult is the question. So it is not trivial to do genome editing using RMPs. So you have to really work in the optimization process. But I think, I mean, we have shown that it is possible. If you have lower frequency, you might need to go and then probably bombard a little bit more than other species, but perfectly be doing.
Great. Another question is, are you afraid of making herbicide-resistant plants with CRISPR will negatively influence public acceptance of CRISPR as a technology?
That's a real good question. And this is my call, okay? This is not the company call. So we're not, so we're working, so we have partners, like they are developing herbicides. The idea is this new concept of the greener herbicides, right? So it's not, so the idea would be to use that greener herbicide that have less environmental impact, not only in the environment, but also in crops, and then introgress through genome editing, introgress that to basically bring a new technology in here. So, I mean, thinking about the future, right? So you basically forget about what is out there, like the classical or the glyphosate. Probably everybody is thinking about glyphosate. And now the idea is to bring those greener herbicides here, and then to switch that paradigm, right? But I mean, I don't know how the acceptance could be. I mean, the good thing is that it's not. So we are doing it through a GMO-free manner. There is no any. I mean, if we just produce have one nucleotide change in there, like I show you, it won't be regulated by APHIS or USDA. And in Argentina, we are hoping the same. So.
Great. We'll do a few more questions. Is the CRISPR-induced gene modification detected in all parts of the soybean or sorghum explants?
Yeah, that's a really good question. So what we are doing is we bombard and we let them recover from the bombardment, like because it's a real. So you are basically going through the cell wall and the cell membrane. We let them recover for between 48 and 3 days, 48 hours and 3 days. And then we collect the samples for doing NGS. What we are doing here is that I mean, if you take in consideration that scatter pattern typical from the gene gun, we're trying to remove as much as we can from the wild type cells. I mean, wild type cells, the cells that we suppose did not receive any impact from the bullets. We're trying to remove them and then just extracting DNA from the ones we suppose are receiving the most particles, basically. So, but yes, we are not sending the whole thing, so we are just cutting selected pieces.
Great. We'll do one more question, and
Okay.
it is, did you find a good correlation between in vitro and in plants regarding the activity of guide RNAs?
Yeah, actually, that would be my dream result, to be honest. So the thing is, from the in vitro activity is basically for us is a go no go strategy, right? So the one that if you have a guide RNA and that guide RNA doesn't cut, you will not use it for in vivo, right? So just stay away from that guide RNA. But we haven't see a strong correlation yet between the activity in vitro and the activity in vivo. There are some papers that are describing that, but in our hands, it's hard to say. But probably we'll be able to do it soon because we have a huge, different projects, huge different guide RNAs that we are testing and then trying also the quantification method for the in vitro activity and then see how that extrapolates to the in vivo activity, basically. But that's a really good question.
Okay, great. Thank you. We're going to conclude this session, give you a few minutes since the panel discussion with the producers of Human Nature is starting at 12:30. So I'll give you some time to get into that session. But first, I want to thank all of our speakers for their time and, of course, their presentations. And thank you guys for attending. Once again, I want to remind you that the expo hall is open. There are booths present. Feel free to visit all the booths and Synthego booths as well for your chance to network with others as well as win some free prizes. So with that, we're going to conclude this session. So thank you, everyone. And feel free to hop on the panel discussion, which will happen at 12:30.